morning. So we will see different phases of the aerobic respiration. The first phase is glycolysis. Now this phase is common to both anaerobic as well as aerobic respiration. Simply, the breakdown of glucose molecule into two molecules of pyruvic acid, it is called as a glycolytic cycle. Now this glycolysis is also known as EMP pathway because the three scientists, that is Emden, Merlock and Parnas, they first time discovered this pathway, and therefore this pathway is called as EMP pathway. The glycolytic reaction, it is a cytosolic pathway because it is taking place or occurring in the cytoplasm of the cell. Now here the glucose is a substrate for the glycolysis. The sources of the glucose may be starch, may be lipids, proteins or sucrose, and this glucose has been derived from all these sources after their breakdown or oxidation. Now we will see the different steps of this glycolytic cycle. Now here, as I told you, the glycolysis starts with the glucose. It is a glucose serve as a substrate, primary substrate for this glycolytic cycle. Now this glucose molecule is phosphorylated first into the glucose 6-phosphate. Now here one ATP molecule is utilized for the phosphorylation, and this reaction takes place in the presence of hexokinase enzyme. Now the glucose 6-phosphate undergoes isomerization, and after isomerization the fructose 6-phosphate is formed up. The reaction of the isomerization is catalyzed by the enzyme phosphoglucoisomerase. Now this fructose 6-phosphate again undergoes phosphorylation. It is phosphorylated. So the ATP is again used here. The phosphate is transferred to the fructose 6-phosphate and fructose 1,6-bisphosphate is formed up. Here this reaction is catalyzed by the enzyme phosphofructokinase, and this PFK or phosphofructokinase enzyme is considered as key enzyme of this glycolytic cycle. The one phosphate group is attached to the carbon number first, and another phosphate group is attached to the carbon number 6. Therefore the name is fructose 1,6-bisphosphate. Now this fructose 1,6-bisphosphate further splits into two molecules of trial sugars, and these are 3-phosphoglyceraldehyde and dihydroxyacetone phosphate. Now both these 3-phosphoglyceraldehyde and dihydroxyacetone phosphates, they are interconvertible and they can be converted into into one another as per the need of these plants. DHAP can convert into phosphoglyceraldehyde and phosphoglyceraldehyde can convert into dihydroxyacetone phosphate. So this reaction of isomerization takes place in the presence of this enzyme triose phosphate isomerase. Now the 3-phosphoglyceraldehyde further oxidized into two molecules of 1,3-diphosphoglyceric acid, and here the oxidizing agent NAD is used. So this NAD gets converts into NADH2. So here two molecules of reducing power NADH2, they are formed up. The reaction of the oxidation of the 3-phosphoglyceraldehyde into 1,3-diphosphoglyceric acid takes place in the presence of enzyme glyceraldehyde 3-phosphate dehydrogenase. Now this 1,3-diphosphoglyceric acid is converted into 3-phosphoglyceric acid. Now from the 1,3-diphosphoglyceric acid, the phosphate groups, they are removed and they are directly transferred to the ADP, and there is a synthesis of ATP molecules. So this reaction takes place in the presence of enzyme phosphoglycerate kinase. Here this reaction is also called as substrate level phosphorylation, because in this reaction 1,3-diphosphoglyceric acid acts as a substrate, and from this substrate the phosphate groups, they are removed and they are directly transferred to a chemical moiety ADP, and there is synthesis of ATP molecules. Therefore this reaction is called as substrate level phosphorylation. Now this 3-phosphoglyceric acid undergoes mutation and it forms 2-phosphoglyceric acid. So this reaction of mutation takes place in the presence of enzyme phosphoglycerate mutase. Here this 2-phosphoglyceric acid is dehydrated to form phosphoenol pyruvate. This reaction takes place in the presence of enzyme enolase, and metal ion magnesium acts as a cofactor. Phosphoenol pyruvate is converted into pyruvic acid. This is the second reaction where the phosphate groups, they are removed from the substrate phosphoenol pyruvate and they are directly transferred to the chemical moiety ADP to form ATP. So this reaction is a second reaction of substrate level phosphorylation. The conversion of phosphoenol pyruvate into the 2 molecules of pyruvic acid is taking place in the presence of enzyme pyruvate kinase. So here in the glycolysis, 1 molecule of the glucose is broken down and it results in the formation of 2 molecules of pyruvic acid. The net output of this glycolysis is 2 molecules of NADH2 and 2 molecules of ATP, they are formed. Thank you.